Hi, my name is Ratika, and today we're going to be looking at the accuracy of reported scientific findings in main media outlets in comparison to the primary source articles about which these media sources write about. For this presentation, I'll be talking about Fox News' article listed at the very top and its primary article source, which is listed right underneath. For this presentation, we will first look at the key points of the article written through the perspective of a main media outlet. Second, we'll take an in-depth dive of the primary article and its individual components. Third, we'll take a look at the levels of reporting accuracy in the news article in comparison to the primary article to determine how accurate the reporting of scientific findings was. And lastly, there's a works cited page so you can check out the sources for yourself and see how accurate the article is on your own time. So let's jump right into this article written by Fox News on new gene editing technology. A little bit of background before we delve into all of this. The disorder that's talked about in the article is called progeria. Now, progeria is a genetic disorder that causes premature aging, and according to Fox News' reporting, this experiment focused mainly on addressing progeria. The Fox article first begins briefly by mentioning the new gene editing tool, SATI, that is being used to edit a wide variety of cell types and varying cell mutations. Next, the article moves on to describe exactly how SATI works to edit the gene, talking about how instead of replacing the troubled genes entirely, the researchers use SATI to put a good copy of the gene into a mice's DNA. Next, the article goes on to talk about the results of SATI in the experiment on mice with progeria, concluding that the use of this new gene editing tool led to diminished aging in organs and an increase in lifespan by 45%. Lastly, the article briefly mentioned that this new technology is based on the original CRISPR-Cas9 gene editing technology. Now, let's take a look at the primary Science Journal article. The primary article is separated very clearly into individual sections. First, there's the abstract. The abstract talks about how in vivo gene editing technology is being used for this experiment and how it uses the insertion of a mini gene to focus on the point mutation that causes progeria. Then there's the introduction. The introduction goes into a lot of detail about how SATI is a combination of two older gene technologies and describes how the combination of these two has allowed for the scope of SATI to become much more vast and allows for a greater variety of cells to be targeted and greater amount of mutations to be targeted as well. Next, the article goes into a lot of detail about the results of this experiment. First, it talks about the phenotypic symptoms of progeria. For mice experiencing progeria, the symptoms have often been progressive weight loss over their lifetime and a shortened lifespan. However, post-SATI treatment, phenotypic symptoms were delayed. Figure 1 right here shows that mice that underwent this treatment saw an overall increase in their lifespan. Second, there was also discussion about the manner in which SATI impacted the condition of progeria mice's organs, as progeria causes organs to age at a faster than normal rate. The result can be seen in figure 2, where mice who underwent SATI treatment saw decreases in level of progerin, which leads to progeria, in the organs listed right here in figure 2. Now let's take a look at the accuracy of the news article. Looking over this entire article, there were definitely issues in the reportings of the scientific findings. First, there was an issue with the basic definition and terms mentioned throughout the article. Specifics were often avoided, and as a reader, I felt that I would have to look on outside sources to get a clearer understanding of the various terms that were mentioned. Secondly, there was a lack of data in this news article to support the conclusions that were drawn. For example, the article mentions that mice with progeria had an increase in lifespan by 45%, but it doesn't really mention anything else about how mice saw a decrease in their aging organs or saw increases in their weight, for example. Nothing that would help the audience really understand how progeria affects and how SATI is helping to at least cure progeria a little bit. Lastly, There was no real mention of why CRISPR-Cas9 was modified in the first place to make SATI. The average person doesn't really understand gene editing technology, much less CRISPR. So giving to them this knowledge that SATI is more modified than CRISPR, it probably creates more audience confusion. And overall, that doesn't work out too well. Overall, from a scientific perspective, there was definite and clear issues in accuracy when looking at Fox News' reporting of this new gene editing technology. This is the works cited, and I will have both of these links linked down below in the description if anyone wants to check this out. And yes, thank you for everyone who stayed and listened to this, and I appreciate your time.